Hello, and thank you for joining this OncLive peer exchange titled Precision Medicine in Advanced Non-Small Cell Lung Cancer. Metastatic lung adenocarcinoma remains the model for translational medicine and oncology, as the elucidation of new molecular targets and rapid development of novel targeted therapies continues to transform the landscape of therapy. In this OncLive peer exchange, I'm joined by a group of my colleagues, all leaders in the field of thoracic oncology clinical research. We're going to talk about the targeted therapies that we have available today, the data from the 2018 ASCO annual meeting, and how the latest research will advance the clinical management of non-small cell lung cancer. I'm Dr. Benjamin Levy. I'm an assistant professor of oncology and clinical director of medical oncology at the Johns Hopkins Sidney Kimmel Cancer Center at Sibley Memorial Hospital in Washington, D.C. Participating today on our distinguished panel are Dr. Luder Bashanova, medical oncologist and professor of medicine at UC San Diego Health, Dr. Alex Drilon, medical oncologist and clinical director on the Early Drug Development Service at Memorial Sloan Kettering Cancer Center in New York, New York, and Dr. Zofia Piotrowska, instructor of medicine in the Department of Hematology Oncology at Mass General Hospital Cancer Center in Boston, Massachusetts, and Dr. Jonathan Riess, assistant professor of medicine in the Division of Hematology Oncology at UC Davis Comprehensive Cancer Center. Thank you so much for joining us. Let's begin. So I think we've learned a lot about precision medicine and other tumor types. In my mind, lung cancer has been the poster child for personalized medicine. And I think that story really begins with EGFR. This is a mutation that's relevant and identified in up to 15 to 20% of all non-small cell lung cancer adenocarcinoma. And we now have important phase 3 trials comparing TKIs, either erlotinib, gefitinib, or afatinib, that outperform chemotherapy in terms of response rate, in terms of progression-free survival, and importantly, in terms of quality of life. And that's really altered the way that we think about treating these patients. But we've got now more practice-changing data with a next-generation TKI in Osimertinib. So Zofia, you want to walk us through the story with Osimertinib, landing initially in the T790M space, but then more importantly, the FLAURA data that has been recently published and changed things for us.
Yeah, it's been a very exciting time. Osimertinib was formerly known as AZD9291 and first came on the scene probably about 5 years ago now as a drug that was first developed to overcome T790M-mediated resistance to the first and second generation EGFR inhibitors. T790M is a resistance mutation in EGFR that we know is seen in about 50 to 60% of patients who progress on erlotinib, gefitinib, and afatinib. And with that knowledge in mind, Osimertinib was developed for that indication and was quickly shown to be quite effective in that space. So there was phase 1 and phase 2 data, and ultimately the phase 3 AURA3 study, which really showed that Osimertinib was superior to chemotherapy for patients who were T790M positive after a first or second generation drug. Most recently, actually, the Osimertinib was moved to the first line setting based on the results of the FLAURA trial. FLAURA was also a randomized phase 3 study which compared Osimertinib to standard of care, which could be erlotinib or gefitinib, and demonstrated a really meaningful improvement in progression-free survival for patients treated with first-line Osimertinib with a median PFS of about 19 months compared to the standard of care arm, which performed as expected around 10 months. So this data presented at ESMO last year has really, I think, transformed practice. It has led to the FDA approval of Osimertinib for first-line treatment of EGFR mutant lung cancer just earlier this year.
Yeah, I think we initially thought of Osimertinib, as you've discussed the story, as this great T790M drug, but it really hits quite nicely the sensitizing mutations, which was the rationale for moving it up front. In terms of toxicity differences, that's also a kind of a highlight of the FLAURA. Do you want to comment briefly on just the discrepancy in toxicities between Osimertinib and first and second generation TKIs?
Yeah, so one of the really nice distinguishing features of Osimertinib has been its nice therapeutic window where it's very effective and potent against mutant EGFR, both the activating mutations in T790M, but relatively sparing of wild-type EGFR. And that has translated to a very nice safety profile with decreased rates of the typical EGFR toxicities we expect to see, including rash, diarrhea, skin toxicities. Yeah. So it's been, not only has it a very effective therapy, but also really well tolerated for our patients.
And people's clinical experience with Osimertinib so far in terms of its, I think we all know its efficacy and we're learning about it in clinical practice, but certainly FLAURA cemented it with a PFS close to 2 years, 19 months. But comments on just the tolerability of this drug in everyday practice? Are people seeing, or the panelists here seeing toxicities that to be looking out for, or is it pretty well tolerated across the board?
Overall, I found it to be fairly well tolerated, particularly compared to the previous generation EGFR TKIs with respect to rash and diarrhea. It still can occur, and it still could be substantial enough where you need to take into account a potential dose reduction, but I found it to be a good amount less and very manageable. Other things that I've noted is, there is a small risk of pneumonitis, and typically with the first and second generation EGFR TKIs, we've predominantly seen that in a more Asian population, but I found that there's been some data shown that this can also happen with osimertinib in more of a Caucasian population as well. So it's something not to discount, and it's something that that is uncommon, but we do see and to keep an eye out for, including occasional kind of patchy infiltrates that may come and go without any clinical significance, but sometimes it could also be more substantial. So I think the one takeaway message in terms of toxicity is that's something that's uncommon, but I do keep a careful eye out for.
Yeah, the last takeaway message I wanted to throw in is the fact that we're really getting better at designing these drugs that not only have activity against resistance mechanisms, but also have excellent CNS coverage.
Right.
And osimertinib is certainly one of these drugs. It's been shown to work in patients with brain metastases, but beyond that, in the Bloom study, we also saw that it can work in patients with leptomeningeal disease.
Yep.
So a big win for patients, I think.
I would agree. I mean, the PFS curves held up in those patients with brain mets enrolled in this study, which was not a small amount of patients in this trial.